Yes, ma'am, we are live. You can start, ma'am, please.
Okay.
Yeah.
Shall we start, sir?
You can start, madam.
Yes, ma'am.
Okay.
Yes, ma'am. You can start.
Yes, sir. Thank you. So, good morning to one and all. I'm Dr. Gandhimadi. On behalf of Teaching Learning Center of Ramanujam College and Department of Zoology, PSGR Krishnamal College for Women, I welcome the resource person and the participants for the second live session on animal model for biomedical and toxicological studies to be delivered by the Dr. Gerald Mahesh Kumar M, Senior Principal Scientist, Department of Biotechnology, Center for Cellular and Molecular Biology, Hyderabad. Now, I may request Dr. P. Sushila, Assistant Professor, Department of Zoology, PSGR Krishnamal College for Women, to introduce the resource person to the participants. Over to you, ma'am. Thank you.
Thank you, Gandhimati. Good morning, all. I take this opportunity to welcome our guest speaker and the participants for the third day of the online refresher course program in zoology. It is a moment of extreme pleasure to introduce the speaker of the day, Dr. Gerald Mahesh Kumar, Principal Scientist and Officer in Charge, Center for Cellular and Molecular Biology, Hyderabad, who has joined us today. Dr. Gerald Mahesh Kumar completed his bachelor's and master's in veterinary science with a specialization in veterinary pathology from the Tamil Nadu Veterinary Animal Sciences University, Chennai. He has received the most prestigious Japanese Laboratory Animal Science International Young Scientist Award from the Japanese Laboratory Animal Science Association in 2007. He obtained his doctoral degree from the School of Pharmaceutical Sciences, Kumamoto University, Japan, for which he received the Ronpako Fellowship in 2008 from Japan University for Promotion of Science. After completion of his PhD, he worked as a lab animal veterinarian for the period of 4 years at the National Institute of Immunology, New Delhi. He is associated with the Center for Cellular and Molecular Biology as a principal scientist for more than 17 years. His research mainly focuses on the role of various antioxidants and their mechanisms on anti-atherosclerotic properties in ApoE knockout mice. Sir has completed 10 research projects funded by the ICMR and DBT as principal investigator and co-principal investigator, and he has published more than 113 research articles in peer-reviewed international journals. With these few words, I once again express my heartfelt thanks to Dr. Gerald Mahesh Kumar for accepting our invitation in spite of his busy schedule, and therefore your deliberation will surely benefit the teaching fraternity. I now invite Dr. Gerald Mahesh Kumar to share his expertise to enlighten the participants. Thank you once again, sir. Thank you.
Thank you, madam. May I audible?
Yes, sir.
Yeah. Okay. So most of the participants are the students and faculties, or
Only faculties, sir.
only faculties?
Yes, sir. Professors from various colleges across the country.
Okay. Professors. Okay. Thank you. Yeah. Can I start?
Yes, sir. You can start, sir.
Okay. Yeah. So my topic is animal model for biomedical research and toxicological studies. And in this presentation, I want to give about what is the introduction part, what are the histories of the breakthrough achieved through the animal research, and third part, why animal research is necessary. Is it required for animal research? Now, so many alternatives are coming out, and cell cultures techniques are coming up, and why this is really the animal research is necessary. And we are going to discuss and different type of animal usage in biomedical research. That means we have a different area of research. It may give some kind of experience what kind of area they are using for animals, so stem cell research or transgenic research or pharmacological research or vaccine research, how they are utilizing animal and what purpose they are using the animal. That I want to explain. And again, the source of animal model, and where is the different kind of sources in India or abroad. And finally, we are coming to conclusions. What is the role of animals in biomedical and toxicological studies? You can see them. This is a rough estimate this throughout the world, throughout the globe, they are using 80 to 100 million, maybe cross more than that also. This 80 to 100 million, what we are starting from lower vertebrates, the lower vertebrates starting from zebrafish to the up to the large animal. Large animal, the people they are using dog, they are using, and sheep and goat and non-human primates. These are all the widely used large animal model for various drug screening programs for prior to the clinical studies. And the major animal model where they are, who is using the most animal model, you can see the mostly the pharmaceutical companies. Pharmaceutical companies also two different purposes. One is purely drug discovery program, and another one is contract research laboratory. If any drugs molecules are developed in the R&D lab, then again it need to go for a regulatory requirement. This regulatory requirement, you know that, everybody know that. All the drug and pharmaceuticals and cosmetics, what else are coming into the market, after 2020, the Indian regulations, DCGA framed the regulations, all the drugs, all the pharmaceuticals, all the vaccines, all the cosmetics that need to be tested under the GLP lab. That also now earlier day contract research lab, you can do any kind of research. Even contract research lab in small lab, they have animal facility, they can do all kind of research activity. But now after 2020, all the drugs need to be tested under GLP. GLP means Good Laboratory Practice Lab. In India, 46 GLP certified labs are there. 46 certified. This Indian GLP, the Good Laboratory Practice Lab, are accepted by the throughout the globe. OECD accepted, US FDA accepted. Like many countries are accepted. If the drugs are tested in India, no need to retest again for preclinical trial in anywhere about the world. Directly you can go for clinical trials to market to get the regulatory approval. That's very, very important. What is the purpose they are doing for drugs and cosmetic? Mainly, all the drugs need to go for preclinical trial. Preclinical trial, how the various battery of tests are there. The preclinical study, it is what they are doing. They are not only doing for toxicity. They have to first study them safety. The safety is very important. Then they will check them potency. They will check the efficacy, and finally they will check the toxicity. These are all the basic things they have tested in the animal model. And if they have different battery of tests are there, that if you are testing all the kind of battery of tests, if you are cleared there, then you can submit to these regulatory, specifically India regulations are governed by the DCGA. The DCGA, they will give approval for preclinically if the drugs are regulated and approved, then you can go with the clinical trial. If their clinical trial have also phase 1, phase 2, phase 3 trial. If you are depending upon the drugs, sometimes they will go with the phase 1 and phase 2. From there, they will give for emergency approval. Like you can see the COVID vaccine. The COVID vaccine not go for phase 3 trials. It's reach up to phase 1 and phase 2 trial. Phase 1, phase 2 trial. From that, the clearance. The phase 1, phase 2 trial, it will be this the maximum 5,000 population. If you are phase 1 trial, 200 human population. Phase 2 trials, 5,000 populations. If you are phase 3, there will be lakhs. That will be possible, not possible, such an emergency situation. The people, the DCG regulation, they are approving on emergency basis, requirement of the public, very urgent nature. That's the COVID vaccine. Like this emergency nature, many drugs are approving, the works are going on. Such a way, the regulations are developed. But even we are going for emergency approval also, you need the drugs or vaccine need to be cleared from the preclinical. Preclinical set of parameters. That is why the preclinical regulation will be regulated by the DCGA. For that purposes, huge number of animals required for the industry. And earlier, any one contract research laboratory can do. Now the systems has changed that need to go for GLP lab. Even in Tamil Nadu also, they have few GLP labs are there. You can see them. Chennai have GLR lab, Bioscience lab, both are GLP lab. Again, this is Pipette. Pipette also is one of the leading GLP lab in India, and few of the organizations that are also coming up nowadays. That's very, very important for the GLP lab now to test for all kind of drug and cosmetics. And apart from that, what are the animal model they are using for research laboratory? Research institute, most of the research are exploratory. That means they are in the basic research or they are applied research. That will not be directly applicable, but they are doing the research. They have to publish the paper. Based on the publish, the report, the industry will take the data and they will plan the experiment for regulatory purposes. That's a very important. Mostly the institute levels, all the government institution or private institution, Most of them they are doing for exploratory research. That also we record large number of animal. Again, defense research laboratory, that also they are using large number of animal, but we are not aware the data will not come up. But apart from that, you can see them 80 to 100 millions of animal throughout the globe we are using them. And you can go with them. What are the major achievement? You can see them breakthrough, how it happened with the animals. Just I am few things I am putting. It's so many advancement happen using the animal. Very many day to day we are using the drugs and developments, surgery, many kind of advanced surgery that are all developed by using animal. Unless until the usage of animal, they can't establish and standardize. That's one of the just a few clippings I put there. Maybe that for your references. You can see the insulin. The insulin was, I put on insulin discovery. I want how they are insulin discovered by using the animal. You can see them. There's a 3 students. They are medical student. They are joined in Canada. So you know that Bank, McDonald, and there are 3 students. They are this is a medical student, graduate student. They are doing for MD program. For MBBS, they are doing for MD program. This class is what we are suggesting them. We are giving 10 dogs. You have to what kind of experiment you want to do. We can do. What they are seeing them, they're at the time of releasing of the journal, they are seeing them, they are find them some kind of secretions are secreted by the pancreas that you play a major role for regulations of diabetes like this. Then this is not a concrete any kind of experiment has happened. It's a report. I said isolates of Langerhans discovered that the Langerhans discovered the pancreas, specifically isolate cell that was published at the time. They never know about what is the roles and Lang. The Pang and McDonald, what they are doing them, they are given the 10 dogs. They are isolating the pancreas from the animal. At the time I identified this is pancreas, their specific functions are not identified. They are doing, and what they are after removing of the dog, the pancreas, what is the symptoms are happening? They are watching them. It's a medical student and they are using animals, they are watching them. They are typically finding the typical symptom of diabetes symptom. The animals are polyuria, multiple urinations, frequent urinations. Again, they're drinking a lot of water, thirsty, and blood glucose level reaching very high. And they are surprised. They are surprised. What we can do? This is a very good model for diabetes. What do you want to do them? What something specialized systems, something specialized chemicals are inside in the pancreas. They thought of the student what they have done, but they do not have the advanced technology. What they are themselves, what they are thinking that they isolated pancreas, they are homogenizing, they are doing them, they are doing the filtering them, they are getting the secretions. The secretion again they are injecting back to the animal, Nowadays we have find them recombinant insulin. Recombinant is a very advanced technology. They are developing through the lab. They are advanced insulin developed. But earlier we have no such technology, molecular biology. What they have done very interestingly, they make them filtration of the secretions of the pancreas homogenate and they are injecting to the animals. Animals are recovering back to the coming to the normal. This is a very surprise innovations. And because of this invent, they have the 3 students, they got a Nobel Prizes. Maybe you are aware that such kind of happening because of animals. That's why I'm telling you the animals play a major role in many diseases. Now, this is the diabetes. Again, you can see the vaccines. You can follow earlier days, 1950s, as a very dreading diseases. Many people died because of polio encephalomyelitis. Nowadays, polio is most of them eradicated. It comes also people coming under the paralysis only in more countries. But what happened in the earlier days? We are the extensive damage of spinal cords, poliomyelitis. The people will die of. Many people died of. I don't know how the virus. The behaving to the animal, we never know that such a situation, what they have done. You can see them first, they have isolate them and bringing to the virus into the animal model. What they have done, they have what's the human dead patient, the dead patient from that, they have take them spinal cord, homogenate, they are injecting to the animals, mice or primates, they are injecting the things, and the animals also showing similar kind of phenomena. And from that, they do not bring to the cell level. If your cell level comes, then automatically that you can make them inactivation and make them to the vaccine will be possible. But with the early day, they are never know about that. Only the source is animal. What they have done, the animal they are passaging the virus. The passage the virus how it is then the form. This is the homogenate of the dead Persian spinal cords, and they are injecting to the animal. Animal also showing similar kind of symptoms. Again, from that they are making to the inactivation of the secretions. And from that, they are used as a vaccine. You can see Salk vaccine. This is the first polio vaccine developed with the Salk vaccine. How he developed this? The viruses are replicating into there's a large animal to the mice, from the mice again coming to the cell culture level, then he is inactivating using the formaldehyde. It's coming to these vaccines called polio vaccines of Salk vaccine. It's a familiarized to save the millions of people in 1950s, 60s. That is why this kind of things are achieved unless until the animals are properly the times utilized. The animal is a medium to enhance their R&D activities. Nowadays, what we are lavish life, we are using them. Unless until they have not utilized the proper purposes, the science will not progress at this level. Not only heart bypass operations, heart lung surgery, they have developed in the animal model, specifically the pig and dog. They have developed them heart lung machine. The heart lung machines are developed, and then they are validated in the animals, and then they are going to use for this human. This kind of things are developed unless until the animals are there, we cannot achieve. You can see the 94's advanced developed transgenic technology. Through the transgenic technology, nowadays so many animal models are developed. What is transgenic? Transgenic, the new gene insertion to this into the existing genome. How they are developing the genome, the most embryo, they are injecting them the gene of interest, and their transmission of the gene into the embryo. That embryo they are transferring to the fostering animals, and again they are characterizing them whether the transgenes is inserted. If their transgene are inserted, what kind of function, what kind of phenotypic changes, and some of them recombinant homologous recombination approach. They are what they are again knockout technology developed. Knockout technology, homologous recombination 98. They have developed, that is also similar way. The gene functions are going to completely lost. What they're making them the advanced gene editing tool. They are developing the recombination technology to silence them, specifically the gene, and what kind of functional defect happening in the animal. Like this way, so many number of animal nowadays they are developing, and that will be very unique to the human diseases. That's very, very important. If you are not if you have established the animal model, then you can test any kind of the drug. These are all exploratory R&D purposes. You can create the model by using transgenic and knockout approaches. Nowadays in knockout also, transgenic also, early days, this is very struggle to go with them. You have to create them.
Excuse me, sir. Sir, you have muted
Okay.
yourself, sir.
Okay, sorry.
Yes, sir.
Yeah. Yeah. Just now I muted or long back I muted?
A few minutes back, sir.
Okay, sorry. Yeah.
Yes, sir.
Yeah. So what I discuss about transgenic and knockout technology, that's very revolutionized period nowadays. The transgenic and knockout technology, the latest one is the gene editing tool by using CRISPR-Cas technology. They are developing different model specifically to the different kind of human diseases, and that is one of the important technology revolutionized by using animals that we can discuss later. Okay, and can go for the why animal research record. Then I asked them, there's some certain specific record. Only the first is R&D purposes, second one is regulatory purposes. You can see any kind of that. Still, why you are already established system, many drugs are developed. Why should you use animal again? This is very in animal ethics point of view. Many people they will ask, but still the many diseases, the emerging and re-emerging diseases are very common nowadays. You can see them. COVID came, multiple mutant came, like Ebola came. Like many diseases, we are not aware that when the virus will behave, how it will behave, how it will be affect the human being. We never know that unless until we do not have a good biological system to study the disease, unless until you cannot disease the disease, unless until you cannot develop a good model for animal, you cannot develop a drug. That is very important. Why so much of time to develop vaccines for the COVID? Because no specific models are available. Because COVID, you can show this, the COVID viruses need to be interact with the ACE receptor. The ACE receptor normally acts in many of the animals. Specifically, you can mice do not have AC receptor, rat do not have AC receptor, hamster have. So that means certain specific animal have certain specific receptor. That receptor can find to the viruses. Then only the virus will be induced the disease in the animal. Similarly, then only the model can be established and can be studied for this any kind of what is the efficacy of the drug, what is the safety of the drug that can be evaluated possible. That's the main thing. The establishment of model is necessary, and emerging and re-emerging diseases are day-to-day life is coming up. That's why very mandatory. You need to develop animal model. That is why we need a more number of animals. Again, regulatory authority, that will be very, very important. If you are releasing vaccine, every batch wise, we need certain kind of test need to do. If your batch have any contamination will be happen. That is why what they are saying, you can batch test, you can see at least 2 abnormal toxicity. What is abnormal toxicity? They are injecting the drug or injecting the vaccine, just a single dose they are injecting, whether the temperature is rise up in the animal. They will check them. That is called pyrogen test. If the pyrogen test if it is pass or fail, that is very, very important. The DCGA, they will ask whether it is pass or fail in the abnormal toxicity. That means if you are injecting the drug, the temperature is elevated above 0.5 degree, that the drug is failed. That cannot be go further. That means every release of batch of drug that need to be passed the pyrogen test, that means not with the drug, cannot elevate the temperature because of this drug. And again, abnormal toxicity. Abnormal drug, they are injecting the drug, they will absorb for 7 days or 14 days. Do you have any kind of abnormal toxicity, abnormal clinical sense is there? Do you have any kind of abnormal behavior is there? Such kind of thing you can note it down.
Excuse me, sir.
Sir, ma'am.
Sir, are you presenting, sir?
Yeah.
Your presentation is not visible to us, sir.
Not visible? 1 minute, madam.
Yes, sir.
I will close my presentation, then I will again share them. Okay?
Yes, sir. Yes, sir. Sure. Thank you, sir.
Yeah. I close my presentation now. I will share again. Share where it is coming. Share. It's opening now. It's opening now. Is it open now?
Yes, sir. It's open. Yes, sir. It's open.
Please, anything is in between, any problem is there, immediately call me. No problem. I have no
Yes,
issue.
sir. Yes, sir.
Huh?
Yes, sir. Sure,
Okay.
sir. Yes, sure. Thank you, sir.
The animal research is mandatory because of mainly the regulatory purposes. This is the way I explained them. What is abnormal toxicity? What is pyrogen test? Such kind of things you record for every batch release of either the drug or vaccine or any kind of recombinant product or small molecule that need to be recorded for every batch release test. That is why the huge number of animal is necessary to do clear the regulatory for the drugs into the market. And what kind of animals, what we are going for using for biomedical research, you can see them. If you are, this is maybe the old year, maybe this in 2020's data, you can see them. The mice is the most common usage of animal, because why they are using mice more? Mice have the most of the human genome is most 99% resemble with the human. That is why the mice, they are using again the mice, and rat also 25%. Rat mostly they use widely used for toxicological study. And rabbit also they are using for raising different kind of antibodies, monoclonal antibody, polyclonal antibody, rabbits and guinea pigs they are using. Hamster nowadays they are using more. This hamster is a very good model to do for COVID research activity. And primates and dogs are India is very limited facility. And again the different kind of research activity, the regulatory purposes and exploratory purposes. The primate is a very good model to study for reproductive toxicity. And dogs also widely they are using for pharmacokinetic study, pharmacodynamic studies. That's why different kind of animals they are using. This is just a percentage, the widely used animal in Indian populations. You can see them again, mice is we are saying more than 50% of the animals they are used by mice. Mice again, 2 types of mice are there. One is outbred mice and inbred mice. This outbred mice is extensively used. Mostly the vaccine research, the diversity of the population is required. That's why the mice is, if you are going with the breeding and inbreeding, the homozygosity will be established. Then that will be go for any specific exploratory basic research activity, that will be the inbred mice is more helpful. But if you are going for outbred mice, the wide extensive study of vaccine research or wide extensive study of toxicity study, because again the drug is going for the heterozygosity of the population throughout the globe. That is why you need to go more widely used for the outbred strain, like Swiss albino mice. Swiss albino mice is widely used for that. Swiss albino mice. Again, CD1 mice, these are all the mice will be extensively used. But in case of, if you are homozygous population, you can go for the C57 mice, is widely used for the diet-induced obesity, such kind of thing you can use for that. Again, the transgenic and knockouts are very limited. They are this very expensive mice. Again, this is the corporate regulatory, they are widely using for the specific transgenic knockout model for their drug screening programs. Again, how many animals? 84% of the animals are using for pharmaceutical and research development, and fundamental research only 7%, and safety testing for food additives, and food also there will be separate regulation required, and that also will be setting around 7%, and other toxicity 2%. This is the major usage of animal different purposes. Again, what purposes they are using for the testing of the compound, and either the vaccines or sera. What is sera? Sera means you can know them. This is many kind of the diseases. Just an example, I am telling them the snake venom. Snake venom. This is if they are the snake bitten by anyone. You have to go for only. You can go to the government hospital. You can get them anti-sera against the snake venom. Anti-sera against the snake venom. How they are getting them? This is very critical situation. What they are doing them. There's the low virulence of the snake. This means they have normally the Indian snakes are classified in 4 categories. One is viper snakes, this is neurotoxic, viper snakes is hemotoxic, and again cobra is neurotoxic. Like a snakes venoms are classified, and they have combined them all the 4 snake venom. This is how in all over the India, the Irula is located in Chennai. They are supplying the venom to the all over the India, specifically those who are production in the hyperimmune serum against snake venom. What they are doing them? They are combining them different variety of the snake's venoms. They are making in a low virulence and injecting to the hearts at different time bond. They are developing them against this venom. Against the venom, they are developing the antibody, and they are against this antibody. They are purifying them. That antibody will be directly given to the patients to save the carrier. Like this. This is a production of sera. That is, you need to test them. The snake venom is whether it is neutralizing against any specific. What they will do, they will be neutralization test. That means challenge study. They have to expose to the low level of venom, and again they are what else? Their sera developed from that animal that will be tested them whether animal survived or dead. They have to test them. That is kind of studies called neutralization study. Every batch of snakes, anti-snakes will be released. We need to test it for the animal. That is why neutralization study. These are all very tough tests. Using more number of animals. Not only snake venom. I am telling. So many products are there. Whether you are aware or not, there diphtheria, tetanus toxoid. You can go for many kind of human disease. If you have the rabies also they have many companies. They are developing them. The plasma product against the rabies. How they are developing this rabies antigen? What you are injecting? Not a rabies virus. Because rabies antigen they are injecting to the equine cars. Why you are using cars? Cars you can get them large volume of serum. That means you can get them at a time. You have to 1 to 3 liters of plasma. You will get them. Then you can purify them. You can large volume of plasma product that you can neutralize them with a rabies virus or tetanus toxoid or diphtheria or snake venom. Such a many kind of products are coming up. That kind of the product that need to be tested whether it is safety. First thing, whether it is the capability of neutralization is there, whether how the potency is there or not. Such kind of tests you need to be carried out. You need a large number of animals. Okay, the toxicology and safety and potency is very important parameter. Using huge number of animal in industrial different purposes, not only vaccine, not only the drug. Apart from that is plasma products also. And what kind of animal? Coming to this, what type of animal models are there? Only spontaneous models are there and experimental models are there. Spontaneous models mean this animal they have naturally they have the mutations happen in specific genes. You can see them, the diabetics mice, you can see them, some of the animals have the leptin gene knockout. This is spontaneously developed. The leptin gene have the mutations. Automatically the animal is developing the higher level of the glucose level, showing similar kind of phenomena of diabetes. Such a models are spontaneous model. That means naturally it's happened, they induce the genes. That is, if you are developing more number of generation by generation, you are going with the inbreeding, multiple generation, more than 50, 60, definitely the background strains of the gene have changed, automatically it's showing some kind of phenotypic variation, but you need to be characterized, then it will be a very good model to study the human patterns. Like a hypertension, SHR rate, this means the hypertension rate, SHR gene have the mutated, it's happening spontaneously. Again, you can see anophthalmia, even we have developed anophthalmia rats. It's mutation in the RX gene, it's happened. We have identified that continuously the every generation, the animals have either the one eye blindness or the both eyes or blindness will be happen. Such kind of things are happening, a spontaneous thing, but you have to validate and you have to publish, then you can go for any kind of the drug screening program. Again, experimentally induced model, you know that even the people who have such kind of models are not available. They experimentally, they are inducing most of the college level, pharmacy college level, and they are the academic college level and life science college level, zoology people. What they are using them, they are in PhD or MSc study, they are developing the diabetes by using different kind of chemical, either the aloxan they are using, either they are using streptozone, they are using different kind of chemical they are using, and again they are establishing with the model for acute and sub-acute. That means they have the single dose, they will be established some complete damage of the model. What is a single dose? Completely the pancreas will be showing the inflammatory changes, animal immediately will be developing the diabetes and gradually developing that will be the type 2 diabetes. That means type 1 and type 2 diabetes is very important. How do you model create them? You are going single dose or multiple dose that will be developed them. Single dose will be the type 1 diabetes and multiple doses establishing for the type 2 diabetes, very common in all over the world for this adult age population. That is the way you have to go for the chemical induction of the model. And again, atherosclerosis. Suppose atherosclerosis is any kind of cardiovascular disorder, you want to study for them, you need to go with hyper diet and cholesterol, high cholesterol diet. That means you have to go with the 30% fat or 45% fat or 65% fat. And again, you need to go for multiple literatures are available. You can go with them. How long you need to do? That's very important. How much percentage of the fat? How much percent? You can go with them. 1 minute, sir. 1 minute. Hello. I will call you later, sir. You can see down the multiple papers are published. You can see the many papers. The one thing you have to very clearly know that how much percentage of the fat they have used. Again, what is the source of fat? Either the animal fat or the plant fat. Again, the cholesterol, how much percentage? Either 0.5% their cholesterol they are adding or 1% cholesterol they are adding. Again, very, very important, how long they are doing, either 2 months or 4 months or 6 months. Again, how they are characterizing the models, whether this, how it is developed, whether this, you are going only checking for cholesterol level or triglyceride level, you are checking them, or you are changing, have any kind of obesity parameter, and or any kind of cardiovascular parameter, what is the ECG, what are the ECG changes are there, such kind of typical characterization, you have to establish the model, then only you can validate the model, then only you can start screening of your specific drugs. And like chemical also, many chemicals they are inducing cancer, and again, this is a long-term administration required for such kind of creation of model. Again, you can genetically modify animals, so many models are available, and you can, this is very, very important, many genes are identified, what are the functions at the levels of molecular level, but the establishment of model, that is very, very important. The same gene, whether it is behaving with the native gene or not, that is a very challenging attitude for the transgenic and knockout technology. That's why we have very good transgenic and knockout lab, and you can see them in CCMP page how many knockout model we have developed. Otherwise, you can search them in Google. So many knockout model we have developed our own. The world is mainly depending upon either Jackson Lab or Charles River. They are only creating many models, but nowadays many labs in India, you can see them CCMB, ISER, NCBS, TAFR, like many lab, they are establishing them the microinjection facility. The microinjection facility is very major requirement for you are injecting a gene of interest into the embryo. If you are injecting the embryo, now this advanced technology of CRISPR-Cas, you design your vectors and design your gene of interest with the desired vectors, and you can injecting the embryo, 0.5 days embryo, then you can transfer to this embryo to the pseudo-pregnant female, then you will get them, characterize them, what does the gene behave, how it behave, and you can study them a long term, and then you can know that what kind of effect on the animal, there's a gene level, molecular level, then characterize, then you will be publish them as a good model for what kind of area, metabolic area, cardiovascular area, that kind of things are possible. Nowadays, there are many Indian scientists are doing that. And coming our model for pharmacology and toxicology is very limited. You can see that we are going for acute study. Normally, the pharmacology and toxicology study, what they are doing, this is a fixed term of study. That means toxicology, you can go for acute study, subacute study, or 90-day study, or 2 years you are going for course study. That means we need a specific type of test, we need a specific animal. That means we need, again, the regulation required 2 species. We are going with mice and 1 large animal, 1 small animal required, 1 large animal required for industrial regulation. That is why very important, your selection of the model is very important for toxicological study. In pharmacology, you have many methods are available. You know, your college level you are doing them. If you are any kind of Arthur's reaction, you can see the albumin you are injecting into the mice or rats, pass. Automatically what happens, this edematous reaction will develop. This type of reaction, Arthur's reaction, like a type 3 hypersensitivity model, you want to develop a albumin. And again, if you want to hamster here, you want to develop, what you can do, you have to inject them like a albumin. Such a chemical model, like pharmacology is basically you are establishing model for specific purposes. Okay. Again, and coming to this other common model, what are the common model? What are the species? What we are normally used, starting from lower vertebrate to the higher animals. You can see them starting with the Drosophila. Drosophila, what purpose they are using? Genetics purposes, developmental biology research activity. What is the advantage? Is very short life cycle. Very short life cycle. What means if you are targeting a scene in this Drosophila embryo in very short time point, you can get what is the result? What is the effect of the gene? That is very important nowadays. They are developing Drosophila and Zebrafish model for cancer model or any kind of what they will do the mutation in the genes and you not need for microinjection facility. That's one of the advantage. The Drosophila embryos and Zebrafish embryo, it will be available. You can see them under the normal microscope. And you can directly inject them. No need to specific setup or microinjection facility. In this academic laboratory level, you can establish them very easier. Drosophila and Zebrafish facility that you can think for as a faculty members. You are sitting here. You can look for no advanced technology like the microinjections and will be the piezo driller. Such advanced equipment not required to inject them in the embryo. Whatever your lab level you are consulting a vector, you can transfer them. Is an embryo of Zebrafish and Drosophila embryo, you can very clearly visible and inject them, and establishing the Zebrafish facility also not expensive. Academic level, you can think it up, do them, and do the work. Researcher can do that in your student, impose them, do that. That will be useful for to study many kind of cancer model creations or any kind of drug screening program. You can validate them. Again, so these are all the common species using for the animal model. And coming to this area of research, what is the major area if you are going for research? Is molecular biology research, nano research, immunological research, and transgenic knockout research, stem cell research, and this is the different area of research they are using the animal. What is the basic research? What they are doing? What is the pathogenesis of the disease? That means what is the basic mechanism of the disease? In terms of, we can say the mechanism of disease. How do you find the mechanism of the disease? How do you find the pathogenesis of the disease? Unless until you have to know them, unless until you know them, a clear indication of at the molecular level. That means you need a clear pathway. What kind of pathway it is involving? What is the disease is involving? Either inflammatory pathway or it is involving with any kind of allergic pathway. That you have to look for them. What is the pathway the disease is behaving? Then only for that purposes you need them a characterization of a disease pattern is at the molecular level. What kind of gene pathway is involving anti-inflammatory or pro-inflammatory, such kind of things. You need to characterize the mice. That is very important. Characterize the animal is required for that purposes. That's why the basic characterization of the animal, basic research, they are people, the basic molecular mechanism, molecular behaviorness of the disease that need for this basic research they are doing them. And again, there will be wide variety of species already discussed. And basic molecular they are using monoclonal antibody and polyclonal antibody. These are basic things you have to pull out the functions of the gene. This very, if you understand, you need to study the function of the disease. You record specific antibody against the specific gene. The specific gene, how they are developing the antibody, either the monoclonal or polyclonal. Polyclonal widely they are using the rabbit. Monoclonal they are widely using for mice. Mice specifically the Balsi mice they are using them. Rat they are using Sprague-Dawley rats. and polyclonal widely using the New Zealand white rabbits. They are using guinea pig also, tongue and cartilage. What are the specific age group they are using for them? Rabbit 6 months to 2-year-old, and mice is 3 months to 1-year-old, and rat is 3 months to 1-year-old, guinea pigs 8 months to 2 years. This is the widely accepted age limit you can use for this raising monoclonal or polyclonal. Again, the very important, not only the age is important, how much concentration of your protein you are injecting. This means the gene regulating with the protein, that's a protein, how much concentration, that is very important, and how you are doing boosting the animal, how you are boosting the animal, primary boosters, secondary and tertiary, and how much concentration. Normally, the recommended is mice is 50 micrograms of protein is required. In case of rabbits, you are using for the minimum 500 microgram level. Then only the first time you are complete adjuvant you are going them, and further you are going with the incomplete adjuvant with a multiple booster, primary, secondary, and again the time point is very important. If you are going with some 0 day, go with the 21 days, you can go with the 42 days, again you can go to 60 days, you can bleed them whether the antibody is raised against your specific protein or not, then you have to validate them. These are all the basic things this required for the animals in the basic research activity. And applied research, mostly the validate the model. What kind of the model established? Suppose you can see the apolipoprotein E, apolipoprotein B. This is the two knockout model. This knockout model, the apolipoprotein G, what is apolipoprotein is regulating lipid neogenesis. In all the body, the genes are involving with the lipid neogenesis. If this knockout of this gene, yes, not in the complete knockout, it's a specific portion of the exon can be removed. If they remove, automatically the behaving of the gene will be different. Automatically, the lipid imbalance will happen, increasing level of cholesterol, triglyceride level, and again this animal, the plaque development in different area, aorta and coronary artery, the plaque development and plaque occlusion happen. Automatically, the animal is similar resemblance model of various cardiovascular disorder similar to the human. These are the basic things because of the involvement of the knocking out of the apolipoprotein E and gene, like LDLR. LDLR also gene is LDLR, low density lipoprotein gene. If you are knockouting them, similar kind of phenomena happen. Suppose you are making ApoB and LDLR, both you can cross with them to make them triple knockout. What automatically it happen? That will be more behaving more cardiovascular disorder models. Such a way you have to create a model. Novel approaches in applied research, you are validating your created model whether the drug is working out or not. That's why I'm saying many kind of the models are developed throughout the world, and we are in the stage of getting them. But now you have to develop the stage of creation of model with the transgenic knockout in the lab level, and that will be improve our Indian scenario. You can see the many different kind of genes. Just I am displaying so many genes are there. DBDB leptin gene, APC, apoptotic coli is a colon cancer, AKR is leukemia, PR7 is a common breast cancer in human, PR7 knockout. This already model created. Like APP genes, this is Alzheimer's Parkinson model, APP, PAX7, PTX3, like many models are there. Even CCM is one of the potential sources, but we can make them in a certain terms and agreement with the Jackson laboratory. Otherwise, you can come and utilize such a, your faculty people are sitting here. You can look for them. Such a kind of collaboration will be possible with us, and like a many CSR lab, they have such kind of animal models are available. CSR lab or ICMR lab or DPT lab, they have such kind of animal models are available. You can impress them, your student, to depute them to do such kind of collaborative activity to improve your scientific publications. Again, this is skid and nude mice. This is a very kind of immunodeficiency animal model. The lack of P cells and T cells and many kind of cytokines. What happened automatically? Now recently developed NSC mice. The NSC mice also there will be lack of many cytokines and NK cells. What happened? That models are complete immune deficiency. That will be very good model. You can humanized mice. You can develop such kind of models are existing in your human. Your humanized mice are there. You can. What is humanized mice? You are creating them similar like a human immune system. Human immune system. We have the completely lack of immune system. Again, your human cells are you are transplanting. All the things will develop. That means your human T cell you are transferring to them, and human T cell will develop. And again, you can challenge you to go for studies. With the humanized mice, you can go for humanized drug trial. You can do. Nowadays, the customized models, customized cancer model are developing to this immunodeficient mice. The immunodeficient mice, you are injecting them a specific can. Suppose the patient have colon cancer. They are suppose they are not responding to any kind of the drug. The hospitals are struggling. This advanced country, what they are doing them. They are struggling with them. The drugs are not working out. What is the drug of choice, they are unable to identify them. What they will do, they are isolating the biopsy. From the biopsy, they are again injecting to the humanized mice or injecting to this immunodeficiency mice, developing the tumor, and they are multiple drugs, they are testing them. Then they are coming out the drug of choice to the patient. Such kind of technology is coming up in advanced country. In India, is very rare. That's why such kind of required for applied research activity. And immunological research activity, what they are doing, can see them mainly they are taking safety, efficacy, and potency. And in safety also, you can see them, I already explained non-specific test, specific test. Non-specific test can go for animal toxicity study, acute, subacute, chronic study, and potentially they are what they are doing challenge study, like I told you, neutralization study. And specific study, they will check for neurobehavioral study and animal weight gain study and specific toxicity. These are all the things. This is immunological. Immunological means any kind of vaccine programs. You need to pass such a kind of test study, then only you can go with any kind of clinical regulations. Again, cancer study, I explained you. Nude mice, there's a immunodeficiency mice, specifically the nude mice, and skid mice, severe combined immunodeficiency mice. Again, now the advanced NSG mice. These are all things are available, and you can check with them if any kind of cancer cell, you can inject them into the xenograft mice, and you can develop the tumors, and then you can study for the longevity of the animals. Longevity means that will be survival of the animal, and again you are studying for the tumor progressions. Tumor progression, you have to evaluate them. Then this is the model we have developed, maybe already published. I don't have lack of time, I cannot explain them. Yes, this is the model we have developed with the nude mice, and we have characterized and we have published, and this model, and from that also we have developed a cell line. Cell line we developed. Now we have cryopreserved this mice embryos as well as the cell lines, and whenever the requirement is there, we can rederive them and we can use for them for that purposes. Okay, and you can see them. Maybe it's wrong. This is 28 transgenic. We have more than hundreds of transgenic mice are developing nowadays in India. Even CCMB alone, more than 40 plus transgenic mice developed. 40 plus transgenic mice. What does the student registration in CCMB? Those who are registering in transgenic facility, we have given them a gene of interest. They have to design the vector. They are the 5 years term. They have to develop the mice, transgenic mice, and they have to characterize the mice, and they have to publish the mice. This is the activity of our transgenic facility. Like this way, many labs are there in all over the India. They are doing such kind of transgenic mice to create the model for human diseases. Automatically, the industry people, they will look for the model. They will take out the research activity and they will test the molecule. They will come up with the collaborations and they will do their drug screening program. Such kind of activities are going throughout India. You can see them. These are all the animal model. We have 72 strains, 72 different type of human disease models are available. Parkinson, Alzheimer, cancer, retinoblastoma, colon cancer, leukemia model, diabetes. Like many models are available because we have the national facility for transgenic mice, but we have some kind of lacunae because of agreement with the Jackson lab. Jackson lab, we are making an agreement whether we are developing mice, we can make a collaboration, but we have them mice we are procuring from Jackson lab or any kind of National Institute of Health. We are Newton Mouse Resources Center in US, we are, and BCCR Australia, like many resource centers are available throughout the globe. But what we are making them, the scientists, they want interested in CCMB, we are making the specific agreement and we are procuring the animal. Certain kind of agreement animal, we are unable to do that, but we can support them. Such kind of collaboration will be helpful to do experiments. Okay. In transgenic activity, the basic things is donor animal. What is the meaning of donor? Donor means you need to give more number of embryos. How do you give them embryo? If you have normal one strain, if you are inbred strain you are giving, it will give embryo 20 or 30. If you are giving for superovulation. Superovulation means you can go with the PMS3 and HCG. This is 2 carbon. You are injecting certain time point, automatically that will be animal will be more ovulation happen, superovulation. Automatically more number of oocytes will release, and you are putting the mating. You are getting next day, you are getting the 5 days embryo, will be more number of embryo will get. But if you are going with the inbred mice, will be less. But if you are going with the hybrid mice, what is hybrid mice? They are crossing up 2 strains. The CBA is 1 strain and C57 BL6J 1 strain. They are crossing the both strain. That will be the hybrid. And the hybrid you are injecting them the FSH or LH. Automatically, the more superovulation, more embryo will get 60 to 70 embryo. That embryo you are what you are doing, processing them, And the microinjection you are doing them, again you are transferring to the CD1 animal. CD1 is the better mothering capability, and again the animals are this normally the CD1, the embryo mother, normally the 13 to 14 pups will deliver. That means it's a good mothering capability. That is why we are using for CD1 as a recipient mother, and again this we are going with CD1 as a pseudo pregnant. What is mean by pseudo pregnant? Animal feel as a pregnant. How do you make them pseudo pregnant? The CD1 mice, this proven fertile CD1 female, what is a proven fertile CD1 female, we are transferring to the vasectomized male. Vasectomized males mean the vas difference are surgically removed, automatically there is an infertile, but the female fertile males you are introducing into the vasectomized male, and when the next day you are taking the vaginal plaque, the plaques are there, animals are feeling that pregnant, that animal only if you are transferring the embryo, the receptivity of the embryo will be yielding higher, more, you'll get the more number of embryos. But then you can go for further screening whether the transgenic is there or knockout are there, such kind of things will be useful for them. Again, stem cell research, you know that same kind of stem cell research, this nude mice are advancedly they are using them. What they will do, they will create the model for them. How do they are creating? Either you have to go with this, if you are liver cancer you want to develop, or if your liver model you want to develop, CCL4 and getting to the Mr. Rat or biopsy, and then again the damage of the liver, then you can transplanting the liver, either surgically you can transplant them. If you are using the nude mice you have to create a model diabetes or liver damage to the animal, then again you are transferring to the stem cells, either this hematopoietic stem cell or bone marrow stem cell, whatever the cells you are, either you have to directly inject to the liver, you have to open, you can surgery, you have to open surgery, you can do, transfer them directly to the liver, or you can go through the tail vein, whether it will be reaching to this exactly the site of cancer development, site of damage to the liver, it will be replaced. What kind of replacement happen? You have to check for them whether this regeneration happen, the damaged liver revert back to normal or not. Such kind of model can be established and revert back. But you have to, the source of stem cell is very important, and characterization of stem cell is very, very important. Whether the potentially regeneration will be happen, such kind of cells only you can go with transfer to the liver damaged animals. We have, these are all the experiment we are regularly doing. We are publishing the paper. Maybe you can refer the papers, but you need a better surgical procedures. You need a surgical procedure. If you are going with thoracic cavity, if you are going with a cardiac surgery, you need a ventilator. Unless until ventilator, you could not do any kind of opening the heart. If you are opening the thoracic cavity, animal will collapse. If you are without ventilator, you have to animal put it in the ventilator or heart lung machine, then only you can do all kind of perfect surgery. If you are going for myocardial infarction model, how do you are creating? We are creating, animal put it into the ventilator, along with the anesthesia will go, continuous anesthesia will go with the isoflurane, inhalation anesthesia, and again we are going to the intercostal space, third and fifth intercostal space, we are making opening, and automatically the thoracic cavity will open, animal normal breathing will be there if you are in the ventilator. Then you are ligating the coronary artery and creating the infarction, the lack of blood supply to the coronary artery, left ventricle region. Animal slowly they develop of cardiovascular disorders, specifically myocardial infarction will develop, necrosis will develop, heart unable to pump, similar like a human cardiovascular death, similar kind of creation you have to create. Such a model creation, after that what you have to do, the stem cell we are transferring to them. Either this intravenously or you are directly again you do the resurgery to animal, directly implanting to them, and what kind of the regeneration capacity of the stem cells that we are evaluating. If we have this, first you have to characterize the stem cells and characterize the animal model, then you can do the saline study, whether how much efficiency of transplantation, how much efficiency of regeneration capacity you have to assess them. This is the nanoparticle. This is also one of the research activity of collaboration with Deccan University, Australia. They are continuously gave them specifically the nanomaterial, nanocases they are coming. We have to establish the model for bone injury. How we are creating, we are selecting the rabbits. Rabbit, we have to do them drilling in the femur bone, and we are implanting them in the specific nanomaterial. How the regenerating capability that we are assessing them. How we are assessing them regenerating capability. You can see them. This is the implanted site of in the rabbit femur bone. You can see them this X-ray. And you have to assess them, the bone mineral density, bone mineral composition by this DEXA equipments. And again, you can go with them X-ray. X-ray can very clearly give them how the bone density, fragile nature, and whether the implant is there or not, whether you have any kind of fracture is there or not, that will give them. Again, you can go with them if you are sacrificing the animal, whether the regeneration is happening, whether this proliferation of osteoblast, osteoclast, you can go with a specific immunostaining, alkaline phosphatase, like a many specific staining to these bone forming cells, automatically whether the regeneration capacity is there or not. You are rather seeing them, black colors are implanting material. This is the implanted site. This is the implanted site. You can see them, the red color area, wherever the red color is, the area is there, the proliferation of bone forming cells. Such kind of things can very clearly visible by using specific immunostaining. Okay, coming to this source, finally 2 slides only. The source of animal model, if you are going for, so one of the best sources is Mutant Mouse Resource Center. This is coming under National Cancer Institute under NIHA. You can, any academic people also, you can take that animal, but you have that, you have them potential resource, potential funding, you have to online make the agreement. This is also very easy procedure, online agreement you can make. Your agreement made, if you are, they are also level stages there. The stage 1, it's normally available. Stage 2, stage 3, there are all cryopreserved. That will be maybe charge will be there. If you are normal one, have no charges, they will give them animal as a free of cost. Only you have to abide with their agreement. That's very, very important. So abide with the agreement. You can collect them only the expensive, you have to bear them transport of the animal. If you are collecting one strain with a 3 or 4 breeding pair, it will be coming around 1 to 2 lakhs. If you have the funding, you can get the model, you can do the research activity. People, you cannot depend upon always the commercial lab like Calci or Taconic. They are hugely, heavily charged, but you are making agreement, only abide with the agreement. What is the abide with the agreement? You have to give them conflict of interest, or if you cannot use for any commercial purposes, you can use for basic exploratory research purposes, and you can always give them thanks to the donors. Such kind of formalities are there that you can make them agreement that will be possible if you are getting grants in your funds. And like a similar MMHRC hard in Japan, and government and non-profit organization also nowadays they are supplying them animals. This is the website. And coming to the conclusions, several medical advances I discussed with the provide evidence by using animals for the purpose of human benefit, but there is no substitute for the living system to study interaction among the tissue and interaction among the organs. Animals are the best surrogate because they are similarity to the human in structure and function and their organ and biological system. Thank you.
Sir, thank you, sir. Sir, shall we go for question session, sir?
Yeah, yes, ma'am.
Yeah.
2 minutes.
So there is one question from the audience. So what is an alternative for animal model?
Alternative for animal model is nowadays in students level their softwares are available, like how their surgery they are doing them. If you are cutting the skin, first skin epidermis, again dermis, again go for muscles. You know that we are opening animal, but the surgically, physically the man in the software technology they are opening them structure. What are the organs are there? If you are opening them, what kind of internal organ? Such a softwares are available. But if you are really, there is a academic purposes. But if you are going for really for the drug screening program, there are a lot of in vitro standardization techniques are standardized, published literatures are available, organ cultures are available, organates are coming up, and these are all the things. Later day it will become alternative, but it will take time. It's maybe 20 to 25 years. Many in vitro labs, many in vitro techniques may be available to the public that need to be stabilized and strengthen the system. Till the time we need animal research is mandatory, but even animal is very, very mandatory for challenge and neutralization study. That cannot be, no one cannot be stopped that such kind of study, no other alternative.
Thank you, sir. So that is what that was only one question.
Not clearly visible.
It is little bit late in the YouTube publication, sir. So
Okay.
So shall we wind up now?
Yeah, yes, you can wind up, madam.
Yes.
Thank you, sir.